We have already studied the structure of DNA, and in that we know that the distance between the two consecutive base pair is 0.34 nanometer. And we also know that the total number of base pair in E. coli bacteria is 4.6 into 10 raised to the power 6 base pair. Similarly, in human being, it is 6.6 into 10 raised to the power 9 base pair. So this is the total number of base pair, and this is the distance between two base pair. So if we multiply this distance by this, we will get 1.3 here millimeter, and here we will get 2.2 meter. So this comes out the total length of DNA in E. coli, and this is the total length of DNA in human being. The size of the cell in case of E. coli is 1 micrometer, and in case of human it is 5 micrometer. Now the question is, how such a long DNA is present in such a small cell? Similarly, in human also 2.2 meter long DNA is present in a cell of size 5 micrometer. So that means that these DNA, they are packed in a definite pattern. So now let us study packaging of DNA in prokaryote and in eukaryote. So now let us see in prokaryote first. In prokaryote, DNA is single, circular, and without a nuclear membrane. Without a nuclear membrane. So now the question is, though this DNA is not bounded by a nuclear membrane, but still it is confined to a particular place. It is not spread throughout the cytoplasm. There are certain proteins, and these proteins are called NAP. NAP means nucleoid associated protein. Because the nuclear region in prokaryote is known as nucleoid. So the proteins which are there, they are known as the nucleoid associated proteins. So these proteins, they help in the packaging of DNA in a bacteria cell. We know that it is a circular DNA. This is further coiled in the form of loops. It is like this. This circular DNA throughout this is present in the form of such loops. And these loops, these are held in position by means of these proteins. These proteins, they keep these loops intact. So that is how DNA is packed in a prokaryotic cell. Now let us see how this packaging occurs in a eukaryotic cell. To be very specific, let us do in the human being. In human, we are taking example of a eukaryotic cell. So in human or in eukaryotic cells, there are many chromosomes, they are linear, and they are bounded by a nuclear membrane. So there is a specific nucleus here. As in case of prokaryote, we have seen that there are NAP, that is nucleoid associated proteins. Similarly, in case of eukaryotes also, there are proteins, and these proteins are histone proteins. These histone proteins, they help in packaging of DNA. These are positively charged. Our DNA is negatively charged. Now again the question, why histone is positively charged? Why DNA is negatively charged? DNA is having phosphate group in this. This phosphate group make DNA negatively charged, and histone, it is having 2 amino acid, arginine and lysine. These 2 amino acid residue, they make histone positively charged protein. Similarly, in prokaryote also, DNA is negatively charged, and NAP, these are positively charged protein. So, histone, they form the core. Histone protein, they form core of 8 such proteins, and 4 are present on this. So these are 8 histone proteins, and 1 histone protein is present here also, like this. Now DNA is wrapped around this histone protein, like this. And the total number of base pair in these coil around this 8 histone protein are 200. So this whole structure, that is 8 histone protein plus 200 base pair, is called nucleosome. So what is nucleosome? Again I am repeating, 8 histone proteins, and these 8 histone proteins collectively we call them octamer. So octamer of 8 histone protein and 200 base pair around it, that form a nucleosome. So such nucleosomes are present throughout the length of the chromatin. Say this is chromatin. There is a chromatin, right? On this chromatin, we are having these nucleosomes throughout the length, like this. So when this is observed under the microscope, these nucleosomes appear as bead, and in between it look like a thread. So what is bead? Bead is nothing but nucleosome, and thread is called linker DNA. In between nucleosome, there are certain stretch of DNA which are connecting one nucleosome to another nucleosome. These are known as the linker DNA. Now further coiling of this structure will occur when this chromatin material has to condense further to form chromosome. We know that when the cell is not dividing, then this genetic material is spread throughout the nucleus, and now this is called chromatin. But when the cell is ready to divide, then this starts condensing and coiling again. So now the coiling of this material will take place further, like this. So now this thread, it get coiled into this. So now where are nucleosome here? Now there are nucleosome, like this. So have you seen how it is condensed here? Further coiling has occurred, and in fact in this again coiling can occur before the final condensation. So ultimately they will form this when the cell is ready to divide. Right? So for further coiling of this chromatin material, non-histone proteins are also required. Non-histone proteins, they further help in the coiling of DNA. Actually, you see here, the negatively charged DNA was wrapped around positively charged histone protein so that this DNA is kept in place, right? And now further coiling is done by the non-histone protein. This coiling of this DNA material is not uniform throughout the chromosome. Somewhere it can be densely, somewhere it can be loosely, then densely, then loosely, like this. And of course, this is having nucleosome throughout this, right? This we have already seen that how condensation of this material has occurred. So this is a densely packed, this is loosely packed DNA. This densely packed DNA is called heterochromatin. And loosely packed DNA is called euchromatin. And when this is stained, definitely this is going to be lighter in color. So this is lightly stained because it is having less DNA in that, and it is densely, so this will give dark stain. Heterochromatin showing less transcription. Sometimes maybe no transcription. On the other hand, euchromatin, this is transcriptionally active because in this the chromatin material is loose. So transcription can occur on this strand, but these are very densely packed. So to transcription can occur. In that case, this heterochromatin has to be converted into euchromatin. All right. So again, I am repeating. Then first of all, histone protein, they are going to form an octamer, and negatively charged DNA get wrapped around this octamer, and around one octamer 200 base pair are there. So these 200 base pair plus octamer is known as nucleosome. When seen under the microscope, it appears like this, beads on the thread, where beads are nucleosome and thread they are the linker DNA. Now further coiling of this structure will occur because when cell division occurs, then condensation of this chromatin into chromosome take place. Now due to coiling of this DNA, further coiling of this DNA, some area of the DNA become densely packed and some area loosely. So the densely packed area, they are known as heterochromatin, and the loosely packed area, these are known as euchromatin.